Good morning, good afternoon, and good evening to everyone. Thanks so much for joining us for our final session of the inaugural Sam Gambier Early Professionals Forum. Today, we have the pleasure of hosting the University of California, San Francisco speakers, Dr. Henry von Brocklin, Dr. Carolyn Guglielmetti, and Dr. Yelena Levy. So for those that are new to this series, it's to honor Dr. Sam Gambier, who was a world-renowned clinician scientist, and visionary who was a leader and pioneer in the fields of molecular imaging, early detection of cancer, and precision. I'm sorry, there's a bit of feedback. Apologies for that. Hopefully you guys didn't hear that. Sorry. And so Dr. Gambier was the Virginia and D.K. Ludwig Professor of Cancer Research and Chair of Radiology at Stanford University. And he was also a fantastic mentor of about 150 postdoctoral scholars and graduate students throughout his career and inspired countless others. And it is in this spirit that we created the Sam Gambier Early Professionals Forum to honor Dr. Gambier and his unwavering dedication to mentorship of future generations of imaging scientists. And so what I'll do for this session is to quickly introduce the three speakers and then hand it over to Dr. von Brocklin to start this session. So Dr. Henry von Brocklin is Professor of Radiology and Biomedical Imaging and Director of the Radiopharmaceutical Research at the University of California, San Francisco. His current research interests include design of imaging agents targeting cancer cell surface markers, development of new strategies for labeling biomolecules with imaging and therapeutic isotopes, preparation of probes for neurodegenerative disorders, and elaboration of zirconium-89 labeled antibodies for applications, including rheumatoid arthritis and HIV reservoir detection. He has successfully translated several of these tracers into humans with over 10 successful INDs for first-in-human studies. He was elected as an SNMMI Society Fellow in 2018, and Society of Radiopharmaceutical Sciences Fellow in 2021. And he's also a distinguished investigator in the Academy for Radiology and Biomedical Imaging Research. Our second speaker is Dr. Guglielmetti, who's an assistant professional researcher at UCSF. And she obtained her bachelor and master's degree in biology and neurosciences from the University of Toulouse in France, and a PhD in biomedical sciences from the University of Antwerp in Belgium. During her postdoctoral studies in the laboratory of Dr. Trammell at UCSF, she developed and validated novel imaging tools to detect inflammation in neurological disorders using metabolic MRI. Under the mentorship of Dr. von Brocklin and Dr. Yelena Levy, she used AraG PET imaging, which you'll hear about today, to detect T-cell infiltration in preclinical models of multiple sclerosis. And our current research is supported by K99 Pathway to Independence Award and focuses on imaging glutamate imbalance in multiple sclerosis models. And congratulations to her. She'll be joining the Department of Radiology at WashU in St. Louis as an assistant professor in the fall of 2023. And our final speaker is Dr. Levy, who holds a bachelor's degree in chemistry from Belgrade University, Yugoslavia, and a PhD in organic chemistry from the Graduate School of the City University of New York. Her training in the molecular imaging field started during her postdoc in the lab of Dr. Sam Gambier at Stanford, where she worked on the design and development of a range of imaging agents intended for different imaging modalities. She currently serves as Chief Scientific Officer at CellSite Technologies, a company founded by Dr. Gambier, and focused on the clinical development of AraG, today's topic, a T-cell imaging agent. And finally, in her role, she is responsible for directing preclinical imaging studies that focus on determining potential clinical applications, as well as providing assistance with clinical trials efforts. And so without further ado, Dr. von Brocklin, I'll hand it over to you.
Great. Thank you, Jason. Thank you very much for the kind introductions of all of us. It's truly an honor to kick off the last Sam Gambier Early Professionals Forum for this year. It was a pleasure to get to know and interact with Sam over the course of my career. Didn't spend a lot of time with him, but was very happy that I did get to know him and grateful that I had the opportunity to do so. Not a week goes by, I'm sure, for many of us that we don't see or hear something that reminds us of Sam and the indelible mark that he's left on our field. And as you'll see in the presentation this morning, that if it were not for Sam, I wouldn't be here giving this presentation. And I have the honor of sharing this webinar with two awesome women scientists. So with that, we'll get started and talk about the development of imaging agent to look at a variety of diseases that's based on the compound called AraG. There we are. So AraG is shown over here on the right-hand side of the slide. It's called AraG because you can see it has a rather lengthy name, arabinofuranacil guanine. It is taken up in cells and specifically T cells and lymphocytes, and incorporated or phosphorylated and then incorporated into DNA. And it leads to actually killing the cell or cell death. Unfortunately, AraG has a very poor solubility. And so it itself could not be turned into an agent that's delivered as a drug. And so it was found that this analog, nelarabine, which has a methoxy group here in this portion of the molecule, could serve as a prodrug for AraG. It is deaminated it's converted by adenosine deaminase into AraG in situ, and then serves to do cell killing. And it's found to be a very potent chemotherapeutic for T cell acute lymphoblastic leukemia, and also for T cell lymphoblastic lymphoma. So it was thought that this agent might make an excellent imaging agent to look at T cells and activated T cells. And it was in Sam Gambier's lab in probably 2009 timeframe that the fluorine 18 version of AraG, it's known as FAraG or today as VisAct, was developed. And here is the first article that was written. Mohammed Namavari was the radiochemist that helped to develop this, as you can see, and others in the Gambier lab. Sam went on to found CellSite Technologies, this was also already mentioned, and they licensed FAraG from Stanford. And Aruna Gambier is and was the CellSite CEO. So this is how I came into the picture because, of course, they wanted to move this forward. They had done a number of studies in the early 2010s and found that this agent looked suitable for translation into humans. But since they had licensed the compound to a company that Sam owned or founded and had family members involved, this created a little bit of conflict of interest if there was to be some funds going from CellSite back to Stanford. So Sam gave me a call. He said, would you be interested in helping us to translate this molecule and help us to work on some of the first in human studies? And that's how we began. The story began in terms of us getting into FAraG imaging and moving it forward into humans. And it's been a great story all along. So we took the original synthesis. We actually automated it. Shown here on the right is a Neptis synthesis box. And we automated this process. It looks like a very straightforward two-step process with an HPLC purification to get to the final product. It turns out that this reaction is quite finicky, only gave 1 to 5% yields. It was about a 90-minute synthesis. And as I said, we automated it, but even automating it, we still had a lot of fun making this compound and making enough of the compound actually to move forward into utilize in human studies. But we did. And all of our first in human studies were done using this reaction. We used it for several years in working closely with CellSite as they continued to work with this molecule in doing some in vivo studies, as you'll learn about, preclinical studies, as you'll learn about later. Over the years, though, and we encouraged the preparation of this molecule by another route. It's very similar in that it was really just some protecting group changes around the molecule here that allowed one to get a more facile synthesis of the FARG. This is a little bit shorter synthesis in time. It's a cleaner product, higher yields, and is very reproducible. And today we now have this automated on our instrument, and we're able to get certainly incredible usable quantities, a couple hundred millicuries of FARG now every single time we produce the material. And so we're grateful for this, and this will certainly help it go even further in terms of use in humans. Well, a little bit about the T cell, about the mechanism of action of FARG. As I've already mentioned, it gets accumulated, or AraG gets accumulated in T cells, and then it gets phosphorylated. And there's actually two enzymes that can do this: the oxidine-cytidine kinase, which is found in the cytoplasm, and a majority of the AraG and FARG gets translated actually into the mitochondria and is phosphorylated by the oxyguanosine kinase. Again, these the triphosphate AraG triphosphate can then get incorporated, as I mentioned earlier, into mitochondrial DNA, or it can get transported back into the cytoplasm, where another enzyme or another protein, the SAMHD1, can actually dephosphorylate the molecule, and then it can get kicked back out of the cell. But it turns out that in activated T cells, this SAMHD1 is decreased, and so you have actually an accumulation now of the triphosphate material taken up into the DNA. And even if some gets transported back into the cytoplasm, it's not going to get readily dephosphorylated, and so then it sticks. And you can see here over on the right a study where using stimulated cells that you can see the increased and retained uptake of FARG in these cells. So it turns out that FARG is very specific for activated T cells. Hence, why AraG was so good at those lymphoblastic leukemia and lymphoma. You can see here that they actually took this is uptake in a tumor here, and they separated the T cells out from the lymphocytes out from the cancer cells, and then they measured the total radioactivity in the CD8s, the CD4s, CD19s, B cells here in this case. And you can see that if you add all these up, that over 70 to 80% of the activity is actually in the lymphocytes and is not in the tumor cells themselves. These are B cells. It turns out that in mice, this gets taken up in a small amount in B cells, but this is not seen in humans. And if they actually took the cells that these T cells that were where they showed the accumulation, they actually had the phenotype of activated T cells, which is low CD62L and high CD44. And so this FACT study showed indeed that these were being taken up, or this mechanism was very active in the activated T cells. I came across this as I was preparing a talk, and it turns out that Sam actually gave a talk at the WMIC meeting in 2015. It was one of the top 5 first-in-human studies that was presented that year. And as the title indicated here, that we have filed for an IND late in 2014, and that IND was actually approved by the FDA in December of 2014, and we did our first-in-human studies in the early part of 2015. And so I thought this was very interesting. I didn't remember that he had presented on this at the WMIC meeting in 2015. Well, here are some images taken from the biodistribution, the pharmacokinetics in healthy volunteers. These are images here in the top row in females and in males from between 30 minutes and 180 minutes across the spectrum here. And you can see here over on the right a number of different organs, and you can see uptake in those particular regions. Heavy uptake in the normal, these are healthy controls, heavy uptake in the liver, in the kidneys, of course, but you can see some other other regions where there's a small amount of normal uptake. Well, in the remaining talks in the presentation, you're going to hear a little bit about some of the potential applications and what we're now studying with AraG beyond cancer. And Elena is going to talk mostly in the cancer. Carolyn is going to talk about her work in the area of multiple sclerosis. But we've actually, and don't have time to talk about it today, but have been looking at AraG uptake in HIV, looking for the HIV reservoir and trying to help understand the development of potential therapeutics for HIV. We've done some studies in COVID, long COVID, and I'm sure that you'll hear more about this in the future. And in the future also some work in the area of cardiac imaging. So without further ado, I'm going to hand this off to Carolyn and let her talk to you about the studies that she's done with AraG and some preclinical models of multiple sclerosis.
Yep.
Oh, I have to stop sharing. The important part.
Let me share my screen. Get the pointer.
Perfect.
Here we go. So thank you very much for giving me the opportunity to talk today in the Sam Gambier Early Professional Forum, and I'm very happy and honored to actually share the stage with Dr. Van Brooklyn and Dr. Yelena Levy. And so today I will talk to you about how we've been using AraG to detect T-cell infiltration in multiple sclerosis models. So multiple sclerosis is an autoimmune disease that mostly affects young adults with age of onset for developing this disease being around 20 to 30 years old. And this is a disease that has no cure. So this disease is being characterized by the presence of lesion in the brain and in the spinal cords that actually leads to motor deficits as well as cognitive disabilities. And so when we actually collect tissue, either from biopsies or most commonly from postmortem samples, what we can detect in this lesion area are the different hallmarks of multiple sclerosis. So this is demyelination, so loss of oligodendrocytes and myelin sheets around the axons. And this very often leads to neuronal damage. And so this occurs actually in a very inflammatory environment. So there is actually activation of the innate immune cells that reside in the brain, which are called microglia. And there is also activation of the astrocyte, which is another brain cell type. And together with this activation, we can also detect infiltrations of immune cells from the periphery. So we can detect infiltration of macrophages, as well as activated T cells and B cells. And so what I also want, if we actually look more closely into the lesions, and especially between patients, what is very interesting is that we can see that there is actually a lesion heterogeneity in active lesions. So we have some lesions that have actually a lot of T-cell infiltration, and we have some lesions that actually have very little T-cell infiltration, and where demyelination is mostly mediated by activation of microglia and macrophages. And so right now, the way to actually diagnose and monitor multiple sclerosis is by performing magnetic resonance imaging. So basically looking at T2 weighted image, we can detect lesions here, as you can see with the arrow in the brain, we can detect several lesions in these patients. And by using T1 contrast enhanced MRI, so basically a contrast agent is being injected, and that contrast agent will leak into the brain when the blood-brain barrier is being disrupted. And here we can actually detect some of the lesion actually shows this enhancement of contrast, showing that there is a leaky blood-brain barrier here. So this is very powerful to be able, as I say, to detect multiple sclerosis lesion. But as we can see here in the graph, it is actually not a very good predictor of clinical disability. As you can see here, the MR activity indicated by the arrows actually do not really correlate to the clinical disability experienced by the patient, and also does not really correlate with the disease burden experienced by this patient. And this is especially true in the secondary phase of the disease, where you can see progressive worsening of the disability, but MR activity is actually decreased. So because of this, actually, we need to develop and validate novel non-invasive tools to monitor the immune and therapeutic responses. And this will eventually lead to an improvement in the diagnosis as well as disease management that will further lead to a better patient outcome. So the compound that we wanted to investigate here is AraG because of its selective accumulation in activated T cell. So I'm not going to go over the mechanism that Henry just talked about in the previous part of this webinar, but what I want to emphasize here is that AraG is being taken up by T cells and it's being accumulated in the mitochondria of activated T cells. And work from our collaborator, from actually Yelena Levy, showed that AraG is actually being taken up by human immune cells. And it's being taken up, as you can see here, by T cell, actually stimulated T cell, as well as macrophages and dendritic cells. And what is actually very interesting, that if we look then at which cell type will retain the label, we can see that T cell actually is the one that retain mostly the AraG when looking at 1 hour post incubation with AraG, while macrophages and dendritic cells will lose a lot of the labels. So then, because of this data acquired in vitro, we actually wanted to investigate whether we would be able to detect T cell infiltration in a multiple sclerosis model in vivo. So the model that we used is called Cuprison, an experimental autoimmune encephalomyelitis model, or Cuprison EAE model. So we decided to use this model because it actually has two different types of lesions that resemble the heterogeneity that can be seen in MS patients. So if we then look here at, so if we collect brain and then perform and try to detect then macroglia macrophages and T cell at baseline, so prior to disease induction, we can see that there is a basal level of innate immune cell in the brain, and we have no T cell. After giving 3 weeks of the Cuprison diet, what we can see that there is actually activation of macroglia macrophages and very little T cell present in the corpus callosum, so in the white matter. And then at week 7, actually 14 days post-immunization with the myelin peptide, we can see that we do still have a strong level of microglia and macrophages present in the lesion. And this time we also have a lot of T cells. So based on this histology, we decided to perform imaging studies, longitudinal imaging at 3 different time points. So prior to disease induction, after 3 weeks of giving the Cuprison diet, and after 7 weeks. And so we performed first MRI, so T2 weighted MRI, as well as T1 contrast NS-MRI. And with this, we actually measured white matter lesion, and we looked for potential disruption of the blood-brain barrier. With AraG imaging, we performed actually a static acquisition, was a 10-minute acquisition, and this was acquired 1 hour post-injection of AraG. And so this is to actually, this was the timing that was first optimized by the group of Yelena Levy, showing that this is the time point with which we would have the highest chance to detect active accumulation in T cell. And then we also performed immunofluorescence at these 3 different time points, looking for T cell and microglia macrophages. So what we found with conventional MRI, so T2 weighted MRI, is that after at the 2 different lesion stage, so week 3 and week 7, we were actually able to detect the lesion in the corpus callosum. And what was interesting here is that while we are able to detect that there is a problem, that there is a lesion, we are not able to differentiate between these 2 lesion types. We then performed T1 weighted MRI. So we injected a contrast agent, which is a gadolinium-based contrast agent. And so this compound will enter the brain when there is a leaky blood-brain barrier. And so what we saw was that there was actually a leaky blood-brain barrier at week 7. And so this actually was pretty widely seen in the brain. So you can detect it here if you look at the red color. And what I want to point out here is that there was still a very wide variation between animals. So some animals showed like almost no accumulation of gadolinium, while others had a very leaky blood-brain barrier. So then moving on to what we obtained with AraG. So here this is an image of AraG PET imaging and CT scan, in which we actually co-registered the corresponding T2 weighted MRI from the same animal. And so that actually enabled us to really delineate region of interest in the brain. So we delineated the region that had the lesion seen with MRI, so in the corpus callosum, as you can see here, as well as region in the cortex and in the hippocampus. And what we found was an increase of AraG only at week 7, so 14 days post-immunization. And we saw that increase in the corpus callosum, so where we were able to detect the lesion with T2 weighted MRI, but as well as in the hippocampus and cortex, which are actually regions where we did not detect any changes using T2 weighted imaging. So then what we decided to do was to collect tissue and really evaluate the number of T cells in all of these regions. And what we found, and then we actually wanted to see if there was any association with the AraG signal that we observed. And so when we actually plotted this data and then performed correlation analysis, we found a very strong correlation between the AraG signal and the number of CD3 T cells in the different brain regions. And because MS patients also develop lesion in the spinal cord, we were also interested in looking whether AraG will be able to detect changes in this region. So we delineated the spinal cord in the lumbar part, as well as in the cervical and thoracic region. And what we found, what is actually clearly evident here on the image, is that a very strong uptake of AraG again at week 7 of the disease. And this was also shown in the cervical and thoracic spinal cord and the lumbar spinal cord. And again, we collected tissue, we stained for T cell, and then we look at potential correlation between AraG signal and number of T cell, and we found again a very strong correlation. So next, we were actually wondering whether we would be able to also detect response to therapy in this animal model. So we decided to treat the animal with a drug which is used to treat MS patients, which is called fingolimod. And we decided to use this drug because it actually works by preventing T cell infiltration into the brain and the spinal cord. And so we treated the animals for 2 weeks, and then at the peak of disease here, we decided to perform MRI, AraG imaging, and collected tissue for ex vivo analysis. So we found that we actually verified first the effect of the drug. So when we performed immunofluorescence analysis, we found that indeed the number of T cells in the brain was decreased following fingolimod treatment. So then looking at T2 weighted MRI, what we observed here was still a lesion in the corpus callosum in the untreated animal, as well as in the fingolimod treated animal. So basically, we were not able to detect any effect of therapy using T2 weighted MRI. Using T1 weighted MRI, we actually saw a very strong effect. So basically, the animal that received fingolimod did not show any sign of active lesions. So here we are not able, but only based on this T1 weighted, we will not be able to detect any lesion in the brain. And so this is most likely due to the fact that fingolimod also acts on parasites, which are controlling the tight junction. And so then with AraG, and again, we merged with T2 weighted MRI, we were actually able to detect an effect of fingolimod therapy where we saw a decrease of AraG signal. And so here I'm only showing the signal that we saw the drop in the corpus callosum, but we also observed similar effect in other regions, in hippocampus and cortex. And then we also then looked at the spinal cord. So this time I masked actually the other organs so you can better appreciate the changes in the spinal cord in between the control and treated and finger treated animals. And here we were again able to detect a decrease of FRG signal in the animal that received finger therapy. And this was in agreement with histological analysis that we performed for T cells. So then in summary, I've shown you that by using FRG PET imaging, we're actually able to differentiate between lesions that have no T cell and that have T cell infiltration in the brain. I've also shown you that we were able to detect the therapy response to fingolimod. And so this is really highlighting the high potential to improve the diagnosis and monitoring of multiple sclerosis patients. And so last, I just want to talk a little bit about like future direction. So what do we want to do next now with FRG? So what I'm planning on doing with FRG is to actually perform studies to really look at disease pathogenesis. So can we actually use FRG to detect lesion earlier? So like right now we look only at peak of disease, but can we detect maybe before any symptoms? Can we maybe detect lesion even before MRI can show us that there is a lesion? I'm also very interested in looking at using FRG to be able to detect whether we have T cell actually activation as well in chronic lesions. Here I also brought back actually the first schematic that I show in the first slide, showing that in multiple sclerosis lesion, there are actually activated T cells, but there is also a number of other cells that are activated and are playing a role together with T cells. And so what I'm very interested in doing is to combine FRG with novel MR and PET methods that we have developed in the lab, for example, to look at astrocyte reactivity, at microglia macrophages activation, oxidative stress, and to really be able to investigate cell interplay in vivo. And I think this will also be very powerful methods to also be able to really decide on which tissue to dissect and then to further investigate using guided transcriptomics. And of course, I think developing all these tools also comes with like how to translate it to patients. And I think it will be very exciting to also translate and perform imaging studies in patients with multiple sclerosis. And so with this, I want to end the talk by thanking my lab members. So my lab is actually the Chamay lab at UCSF, and we are mostly a lab doing MR imaging, so using metabolic MRI to look at to investigate changes in neurological disorders. I also want to thank my mentors, Henry Van Brooklyn and Dr. Yelena Levy, for really helping me carry out these studies. This was my first PET studies, so I really thank them a lot. I learned a lot through them. And I also want to thank all the other people here at UCSF that helped me carry out this study and our funding. Thank you. Oh, and sorry, I also, next speaker will be Dr. Yelena Levy, and she's going to be telling you more about the use of FRG in clinical setting.
Okay, great. Thanks, Caroline. Participating in this seminar series that celebrates Sam's mentorship has a special meaning for me. As you've heard, I've spent many years at Stanford learning from Sam, and now I'm part of CellSight, a company that he founded. In my part of the seminar, I will talk about the use of FRG in immuno-oncology, focusing on the big picture, like Sam often advised, and I will show you how we use FRG to assess systemic immunity. Practically every human disease has an inflammatory component to it. The immune response is an incredibly complex balancing act in charge of maintaining another balance, organism's homeostasis. The disbalance in immune response itself has profound consequences for the organism. From Caroline, we've heard about multiple sclerosis, which represents one manifestation of immune response out of balance. overly active immune response. And I will be talking about the other side of the disbalance, about immune escape, which is a critical step in cancer development. Immunotherapy, which is a breakthrough cancer treatment, aims to repair and restore the system of immune surveillance and also establish long-lasting anti-tumor immune defenses. But unfortunately, the remarkable and durable responses that got us all excited about immunotherapy are seen only in a fraction of patients with solid tumors. This graph, where the blue line represents survival of patients on standard therapy, and the yellow line represents survival of patients on immunotherapy, shows that most people, patients who receive immunotherapy, do not show a survival benefit. This graph also shows that there are patients in whom immunotherapy leads to damaging effects. In those patients, immunotherapy actually leads to a rapid decline and death. Also, because of the unique mechanism of action, the benefit of immunotherapy is seen late after the start of therapy, shown here at the 2-year mark. So one of the major problems in our way of better understanding these phenomena is a lack of predictive biomarkers. Without these biomarkers, we really can't tell who will benefit and who will potentially be harmed by the therapy. And also we can't tell whether the therapy is working or not. It was clear from early clinical trials that standard ways of assessing response to immunotherapy would not be sufficient. Shown here are CTs of 2 patients before and after the start of therapy. And as you can see, they both show an increase in tumor volume while on therapy. By standard response criteria, because of this increase in tumor volume, both of these patients would be considered non-responders. However, these actually represent examples of a pseudo-progression, shown here, and hyperprogression, shown here. 2 types of responses that are observed with immunotherapy, which make treatment decisions really complicated. In pseudo-progression, this initial increase that results from immune cell infiltration actually signifies a good response to therapy. So in this case, the patient would require treatment continuation past this apparent progression phase. On the other hand, in hyperprogression, the increase in tumor size actually does indicate a rapid tumor growth that would require stopping of the therapy and alternative treatment. And unfortunately for this patient, he died about 5 weeks after starting therapy. To address the response patterns unique to immunotherapeutics, the radiological response criteria have been updated. And now they require assessment of the changes in tumor size over time. However, this method allows only a late readout of the response, which would be too late for hyperprogressors or people with severe toxic effects. But more fundamentally, this method is tumor-centric and neglects key players in immunotherapy and its intended targets, which of course are immune cells. There are methods that do focus on immune cells. I'm showing here a method called Immunoscore. Immunoscore evaluates density of CD3 and CD8 cells in the tumor core and at the tumor margin. This method has shown great results in a large cohort of patients in predicting response to immunotherapies and risk of recurrence in colon cancer. Inflamed colon cancers with high immunoscore show better response to checkpoint inhibitor therapy and have lower risk of recurrence. However, immunoscore is based on biopsy, which limits its utility in immunomonitoring. Biopsies, of course, are invasive, but their major limitation is that they're really not telling us the whole story. In this ovarian patient, investigators found multiple metastases, each with a distinct immune microenvironment, and this distinct immune contexture will affect how these lesions respond to therapy. Biopsying any one of these metastases would actually not give us information about the immune response on a patient level. But even if we could biopsy all of these tumor lesions, is it enough to be looking only at the tumor microenvironment? And the answer for us is an obvious no. It is now increasingly being recognized that majority of the T cells that are residing within the tumor microenvironment are terminally dysfunctional cells that are resistant to reprogramming with immunotherapy drugs. And it's becoming clear that a successful immunotherapy can be expected only if it induces de novo anti-tumor immunity at the periphery. So shown here in the tumor draining lymph nodes, T cells get properly activated, which leads to the infiltration of fully functional effector T cells into the tumor bed. PET imaging with the right agent is really well and uniquely well-suited method for assessing this system-wide immune status, with providing a non-invasive information on the whole body distribution of immune cells, not only in the tumor, but at the periphery as well. We at CellSight believe, of course, that FRG is a great fit for this purpose. As Henry briefly pointed out, FRG visualizes functional T cells and can track their activation or their transformation into effector cells. I am using here tumor microenvironment for the sake of simplicity, just to show you exactly which T cells take up FRG, but this will hold true for other areas where you find T cells. So as mentioned previously, within the tumor microenvironment, there are all sorts of T cells, and they're not equally useful for immunotherapy. We can find fully powered antigen-experienced effector cells, shown here as round dark green cells. We can find early dysfunctional cells that can be reprogrammed, shown here as round light green cells. But we can also find terminally dysfunctional T cells, as I mentioned, that resist immunomodulating therapies. These are the distorted green cells. FRG accumulates in well-functioning or reprogrammable T cells, but is not taken up in terminally dysfunctional cells to any significant extent. And this unique characteristic can be of great use in many different aspects of immuno-oncology. CellSight is focusing on assessing FRG's utility in patient selection and therapy guidance, as well as therapy response and evaluation of adverse effects. Knowing that within tumors, FRG accumulates specifically in immune cells and not tumor cells, we asked whether we can use FRG to stratify tumors based on their CD8 status. Essentially, can we, with the help of FRG, non-invasively take a look at these tumors that are black boxes in terms of inflammation, and sort them out based on the inflammation into immunologically hot and cold. We imaged a panel of 6 tumors with different immune contextures, And shown here are cross sections with yellow arrows pointing at tumors. The signal varied between tumor types. Some tumors showed signal in the core, some at the edge, and some showed no signal at all, really closely matching what's been observed in a biopsy tissue. So using AraG, we were able to categorize these tumors into basic cancer immune phenotypes shown here: inflamed, excluded, and desert. But what's more important is that we could profile these tumors in terms of the functional status of their CD8 cells. Across this tumor panel, we found that AraG does not correlate with the total number of lymphocytes present, but with antigen-experienced CD8 cells that express PD-1 and thus the target of anti-PD-1 therapy. The association of the signal with the cells that are the target of immunotherapy is, of course, very significant, as it indicates potential of AraG to serve as a proxy for the anti-tumor T cell potential. The next question we asked was whether we can use AraG to assess immune priming. In other words, in tumors that are deemed not ready to receive immunotherapy because they're not well infiltrated, for example, can we use AraG to observe an increase in intratumoral inflammation caused by agents that are often combined with immunotherapies for exactly this purpose? Standard cancer therapies, chemo or radiation, are now commonly combined with immunotherapy, and we investigated whether AraG can visualize immunomodulation by immunogenic chemotherapy. AraG is agnostic to therapy, so we expect similar results with radiotherapy as well. We tested two types of combination chemo, one shown to cause tolerogenic cell death and the other immunogenic tumor cell death that generates both innate and adaptive anti-tumor response. And we longitudinally followed the treatment, imaging before and then 3 and 6 days post first chemo. Those are panels P1 and P2, and 3 days post the second chemo, that's panel post treatment. The dramatic change in the signal was observed only in mice treated with immunogenic oxaliplatin cyclophosphamide treatment. In support of the importance of assessing the systemic response, the differences between these therapies were also obvious in the tumor draining lymph nodes. In the immunogenic chemo group, the signal in the tumor draining lymph nodes grew over time, reaching intensity peak at P2 right before we see a large increase in the intratumoral signal. So it seems that we are able to observe what I have shown you earlier with the de novo anti-tumor responses, immune activation in the tumor draining lymph nodes, and then infiltration of these activated T cells into the tumor microenvironment. The importance of this holistic assessment of immune status, not only in the tumor, but also in the lymph nodes, was obvious when we performed longitudinal study of mice while on checkpoint therapy. We observed intratumoral differences between responders and non-responders, but the clear separation between these two came only when we included tumor draining lymph nodes in the analysis. We undertake these preclinical studies to better understand AraG behavior, but we are focused on the clinical use of AraG. The clinical doses of AraG are now available at 18 sites, located mostly in the US, but also in the Netherlands, Japan, and Australia. Besides in immuno-oncology, we are actively investigating AraG in other disorders as well. As Henry pointed out, cardiac, COVID, and HIV studies are all performed with our wonderful colleagues at UCSF, and we hope to be showing some exciting results that we are seeing in these areas at some of the future meetings. As Henry told you, the study in healthy subjects was completed. We are expanding these studies to better understand the physiological signal we are observing in different tissues. Our aim to use FRG to assess systemic immunity aligns really well with the exciting development, relatively recent exciting development in the imaging field, and that's the Explorer. We were fortunate to partner with Drs. Negar Omidvari and Simon Cherry at UC Davis and scan several subjects. And you can see the clarity with which the Explorer reveals accumulation of FRG in a number of lymph nodes in the axilla, also cervical and inguinal. The general design of our clinical studies is shown here. We are collecting this is for therapy response. We are collecting 2 scans, one baseline scan before the start and one scan early post the therapy start. We are interested in understanding systemic immunologic processes. So we are analyzing these scans on a whole body level. We have partnered for this with a company, AIQ, that has a software that compares the signal between the pre and on-treatment scan to provide these response maps. And so areas of uptake are matched, and then changes in uptake post-therapy are used to sort these hotspots into response categories: green responding, red as non-responding, and gray as stable hotspots. We analyzed 4 head and neck patients using this method, and there is a expected heterogeneity between patients, but we also are seeing dramatic intra-patient heterogeneity. For example, in patient 1, we have detected 50 different hotspots of different kinds, increasing, stable, disappearing. And we think that this information can be very valuable because several studies are showing that the heterogeneity of response, or specifically the existence of non-responding lesions, to be predictive of a poor clinical outcome. And this is what we are seeing in these patients as well. The patient with only increasing or new hotspots had considerably longer survival than the patient with disappearing or decreasing hotspots. Interestingly, the patient with the shortest survival, patient 1, showed an increase in CD8s and activation markers post-therapy in the biopsy tissue. The response map for the patient shows heterogeneous response in the body, but also within the primary lesion. So this example really underscores the shortcoming of biopsy and affirms the significance of a patient-level immune response assessment. This type of analysis can potentially be useful in patient selection and therapy guidance as well. This is a metastatic head and neck cancer patient with multiple metastases here seen in FDG, but they appear to be immune deserts in FRG. With the help of AI, we evaluated these lesions in terms of inflammation. Blue are cold, red are hot lesions. And you can see that in this patient, most of the lesions were cold. And so the value that we see in assessing these hotspots is to actually be useful in guiding novel combinatorial therapies, which aim to improve response to immunotherapies. So, for example, one of these approaches is direct targeting of metastatic lesions with radiotherapy. Those are intended to generate immune response within a lesion and potentially also to cause the abscopal systemic effect. So nobody knows which one of these lesions need to be targeted with radiotherapy. Should they target cold or hot? And we hope that FRG can provide some of the answers. Finally, the disbalance in response to immunotherapy can lead to off-target immune-related adverse effects, and AraG, being a whole body method, allows simultaneous assessment of therapy response. This is a head and neck, melanoma patient scanned prior to and post just one injection of immunotherapy, and you can see that there is a high increase in the thyroid and in the heart, the two sites that have been associated with side effects. And so the ability to say early how toxic the therapy is would be really very valuable, especially in patients with low or no clinical benefit, which was the case for this particular patient, unfortunately. Those of you who were lucky to know Sam personally probably heard him tell a Cherokee story of two wolves. Sam used it to inspire in people what to me was his one of his defining characteristic. His unrelenting focus on good and positive in things and in people. But this story for me is also about the internal conflict between opposing forces, and again reiterates the importance of balance. And we hope the AraG will prove useful in providing some clues about the disturbance in the immune balance and how to most efficiently and safely restore it. Cell site is small, we are fearless, we have a huge network of collaborators. I would like to thank them all because they're crucial for moving the agent forward. I would also like to thank my wonderful co-presenters and all of you for your attention.
Thank you so much to all the speakers, Dr. Von Brocklin, Dr. Guglielmetti, and Dr. Levy. It was a fantastic overview of everything related to AraG imaging. Fantastic results, both on the preclinical and the clinical side as well. We do have a few minutes for some questions. Please feel free to add your questions into the chat, and we'd be happy to answer that live. And also the video will be available online for those who are present and registered. So actually, I personally have several many questions for each of the speakers, but I don't know if we have time to cover all of it. But maybe we can touch base on the most recent presentation here about the tumor microenvironment with AraG versus the peripheral AraG signal. So Dr. Levy, do you mind commenting a little bit on that, on how to interpret images when looking at the tumor site versus AraG uptake at the tumor site versus peripheral lymph nodes and pre and post immunotherapy, for example?
So we definitely don't have enough time for me to answer that question. But briefly, so we are actually just being cognizant of the fact that the immune system is a system. It's not happening only in the tumor microenvironment, it's the whole body, just like cancer is a systemic disease. So we are trying to actually look at these tumors and the periphery, tumor draining lymph nodes and lymph nodes in general, separately, but also as part of a whole, and analyze it. But for this, I have to stress out AI approaches are crucial because manual analysis will be very difficult for this kind of purpose.
Yeah, fantastic point about the AI part as well. So maybe we can have one more thought before we close the session. We have only about 2 minutes. So I wanted to ask each of the speakers to perhaps comment a little bit on their thoughts related to this, this forum on mentorship and being a mentee as well. So maybe if each of you can comment slightly on that, that would be fantastic.
So I'll kick off and try to be brief because we only have a couple minutes left here. But I think mentorship is one of the most important things, especially in our environment, the academic environment. Besides the science, I think it's one of the most important things that we do and that we offer to help and encourage our mentees to find their way in the field. Not everybody ends up in the academic world. There are many opportunities out there. And it's often difficult, challenging, especially when you're new in the field, new, just young, starting out in trying to figure out which direction do I go? How do I get there? So I think it's really important that we take this time to work with our mentees. And I really enjoy and encourage people to come and talk to me. And I've had not only those that are directly in my laboratory, but I've had many others come to me from courses that I teach, from interactions with other groups. And I always love to take the opportunity to hear their stories, their struggles, and try to provide a way to help them see past some of the things they're going through and find their path and find that, find that, passion of what they want to do and how they're going to get there and do it. And it's great to see when things work out really well for our mentees.
I completely agree with Henry here, and actually I'm not really mentoring people yet, so I'm hoping to mentor really soon. But I've been mostly a mentee, so I can talk a little bit about like what I really appreciate about mentorship. And I think what I find very important is to find mentors. Actually, I would say not one mentor, but several mentors in different area of research and also a different stage of their career. I think you will get like very interesting perspective on your work and a lot of different advice also in terms of like work-life balance, or like how do I need to focus my time now if I want to stay in academia, if I want to move to industry, to really help guide you from day to day. And I think what is also very important with mentors, especially when we're young scientists starting, is to have a lot of encouragement as well and a lot of positive feedback, because sometimes we spend a lot of time in an experiment and then it doesn't work and then it feels like it's the end of the world at that time. And it's always good to have mentors to be able to talk about it and relativate this and then be able to say, okay, things will still move on and will move on in the right direction. I think if you have the right people around you helping you with this.
So my turn, super briefly. I don't think that there's a recipe for how to be a perfect mentor or mentee. I think that for me, mentor-mentee relationship belongs to this special group of relationships where the right fit is extremely important for the successful outcome. Relationships such as doctor-patient, or to keep with the theme of immunity, antibody antigen. Considering the number of years that I spent at Stanford, I obviously thought that Sam was a perfect fit for me. Sam was kind and trusting and brilliant. And something that I truly greatly appreciated was the freedom that he provided for his mentees to pursue their passions and to develop in their own kind of scientist. And as a mentor now, I really am just trying to emulate these qualities.
Thank you so much. Those are fantastic points from each of you. And we are over time. I do see a couple questions. So we will be continuing this in the social lounge. You'll see a link on the left-hand side. Please join the speakers right after we close out at this session, and they'd be happy to answer those questions and continue the conversation as well. And so without further ado, I guess we will end by saying thank you so much to everybody and all the speakers, especially for the whole 2022 series of this inaugural Sam Gambier Early Professionals Forum, which we look forward to continuing in the next series in the near future. And certainly with that, we value your input. A survey will be sent out to each of you. If you don't mind giving us some comments on how you think this series can be better constructed and improved to honor Dr. Gambier and his dedication to mentorship. Okay, and with that, thank you so much to today's speakers, Dr. Van Bachlen, Dr. Guglielmetti, and Dr. Levy. And we look forward to seeing you in the social lounge right now. Thank you very much.